Hello everyone, welcome to ITLS Academy. Our lecture is Fundamentals of Immunology. Introduction. The science of immunology is one of the most rapidly expanding sciences and represents a vast area of knowledge and research. Thus, in a short chapter, it is impossible to deal in depth with its theory and applications. Historical aspects of immunology. From almost the first written observations by man, it was recognized that persons who had contracted and recovered from certain disease were not susceptible, that is, were immune to further attacks. In ancient times, the process of variolation, that is, the inoculation of live organism of smallpox obtained from deceased pustules from patients who were recovering from the disease, were practiced extensively in India and China. The success rate was very variable and often depended on the skill of the variolator. The results were sometimes distressed from the recipient. The father of immunology was Edward Jenner, an English country doctor who lived from 1749 to 1823. He had observed on his rounds the similarity between the pustules of smallpox and those of cowpox, a disease that affected cows' udders. He also observed that milkmaids who had contracted cowpox by handling the diseased udders were immune to smallpox. Deliberate inoculation of a young boy with cowpox and a later subsequent challenges after the boy had recovered with the contest of pustules taken from a person who was suffering from smallpox failed to induce the disease, and subsequent re-challenges also failed. The process of vaccination was adopted as a preventive measure against smallpox, even though the mechanism by which this immunity was induced was not understood. In 1801, Jenner prophesied the eradication of smallpox by the practice of vaccination. In 1967, the disease infected 10 million people. The World Health Organization initiated a program of confinement and vaccination with the object of eradicating the disease. The science of immunology not only encompasses the body's immune response of bacteria and viruses but is extensively involved in tumor recognition and subsequent resection. The resection of transplanted organs and tissues, the elimination of parasites from the body, allergies, and autoimmunity. Autoimmunity is the condition when the body mounts a reaction against its own tissues. Definitions. An organism which has the ability to cause disease is termed as pathogen. Disease in humans and animals may be caused by a variety of microorganisms. The three most important groups being bacteria, rickettsia, and viruses. The term virulence is used to indicate the degree of pathogenicity of a given strain of microorganism. Reduction in the normal virulence of a pathogen is termed as attenuation. This can eventually result in the organism losing its virulence completely and it is then termed avirulent. Conversely, any increase in virulence is termed as exaltation. The body possesses an efficient natural defense mechanism which restricts microorganisms to areas where they can be tolerated. A breach of this mechanism, allowing them to reach tissues which are normally inaccessible, results in an infection. Invasion and multiplication of the organism in the infected host may result in a pathological condition, the clinical entity of disease. We know two types of immune systems, that is innate immune system and adaptive immune system. First of all, we will discuss innate immune system. The body possesses a number of non-specific antimicrobial systems which are operative at all times against potentially pathogenic microorganisms. Prior contact with the infectious agent has no intrinsic effect on these systems. The barrier for innate immune system is skin and mucous membranes. So let's discuss. The intact skin is virtually impregnable to microorganisms and only when damage occurs can invasion take place. Furthermore, many microorganisms fail to survive on the skin surface for any length of time due to the inhibitory effect of fatty acids and lactic acids in sweat and sebaceous secretions. Mucus secreted by the membrane linings of the inner surfaces of the body acts as a protective barrier by trapping microorganisms and other foreign particles, and these are subsequently removed by ciliary action linked in the case of the respiratory tract with coughing and sneezing. Many body secretions contain substances that exert a bactericidal action. For example, the enzyme lysozyme, which is found in tears, nasal secretions, and saliva. Hydrochloric acid in the stomach, which results in a low pH, and basic polypeptides such as spermine, which are found in semen. Phagocytosis. Metchnikoff in 1883 recognized the role of cell types, that is, phagocytes, which were responsible for the engulfment and digestion of microorganisms. They are major line of defense against microbes that breach the initial barriers described above, that we have studied in our previous slide. Two types of phagocytic cells are found in the blood, both of which are derived from the totipotent bone marrow stem cells. First one is the monocytes. Monocytes constitute about 5% of the total blood leukocytes. Many migrate into the tissues and mature into macrophages. Second type of phagocytic cells are the neutrophils. Neutrophils, also called as polymorphonuclear leukocytes, that is PMNs, which are the professional phagocytes of the body. Neutrophils constitute more than 70% of the total leukocyte populations, remaining in the circulatory system for less than 48 hours before migrating into the tissues. In response to a suitable stimulus where they phagocytose material. They possess receptors for Fc and activated C3 which enhance their phagocytic ability. Another group of phagocytic cells are the macrophages. These are long-lived cells found in most tissues and lining serous cavities and the lungs. Other macrophages recirculate through the secondary lymphoid organs, spleen and lymph nodes, where they are advantageous place to filter out foreign materials. So we have studied phagocytic cells, that are monocytes, lymphocytes and macrophages. So now we will see the role of these phagocyte cells and role of phagocytosis. The microorganism initially adheres to the surface of the phagocytic cell, and this is then followed by engulfment of the particles so that it lies within a vacuole, or we can say it is a phagosome within the cell. Lysosomal granules within the phagocyte fuse with the vacuole to form a phagolysosome. These granules contain a variety of bactericidal components which destroy the ingested microorganism by systems that are oxygen dependent or oxygen independent. When a microorganism breaches the initial barriers and enters the body tissues, the phagocytes form a formidable defense barrier. Phagocytes form a formidable defense barrier, and phagocytosis is greatly enhanced by a family of proteins called complement proteins. Here comes our complement system. So we are going to study the complement system and other soluble factors. Complement comprises a group of heat labile serum proteins which, when activated, are associated with the destruction of bacteria in the body in a variety of ways. It is present in low concentrations in serum, but as its action is linked intimately with a second or specific set of defense mechanisms. Proteins produced by virtually infected cells have been shown to interfere with viral replication. They also activate leukocytes that can recognize these infected cells and subsequently kill them. These leukocytes are known as natural killer cells or NK cells, and the proteins are termed as interferons. So we can say that interferons are proteins that works against viral replication. These work against virus and stop the replication of virus. The serum concentrations of a number of proteins increases dramatically during infection. Their levels can increase by up to 100-fold compared with normal levels. They are known collectively as acute phase proteins, and certain of them have been shown to enhance phagocytosis in conjunction with complement. Inflammation. One early symptom of injury to tissue due to a microbial infection is known as inflammation. This begins with the dilation of local arterioles and capillaries, which increases the blood flow to the area and causes characteristic reddening. As we know that inflammation is characterized by inflammation, swelling, redness and chill. Fluid accumulates in the area of injury due to an increase in the permeability of the capillary walls, and this leads to localized oedema, which creates a pressure on nerve endings resulting in pain. Inflammation is a non-specific reaction which can be induced by a variety of agents apart from microorganisms. Lymphokines and derivatives of arachidonic acid, including prostaglandins, leukotrienes, and thromboxanes, are probable mediators of the inflammatory responses. The release of vasoactive amines such as histamine and serotonin, that is 5-hydroxytryptamine, from activated or damaged cells also contribute to inflammation. Fever is the most common manifestation occurs in inflammation. The thermoregulatory center in the hypothalamus regulates body temperature, and this can be affected by endotoxins heat-stable lipopolysaccharides of gram-negative bacteria and also by a monokine secreted by monocytes and macrophages called interleukin 1 or IL-1, which is also termed endogenous pyrogens. Host damage. Microorganisms that escape phagocytosis is a local lesion may now be transported to the regional lymph nodes via the lymphatic vessels. If massive invasion occurs with which the resident macrophages are unable to cope, microorganisms may be transported through the thoracic duct into the bloodstream. The appearance of viable microorganism in the bloodstream is termed as bacteremia and is indicative of an invasive infection and failure of the primary defenses. Pathogenic organisms possess certain properties which enable them to overcome these primary defenses. They produce metabolic substances, often enzymic in nature, which facilitate the invasion of the body. Damage to the host may arise in two ways. First is multiplication of the microorganism may cause mechanical damage to the tissue cells through interference with the normal cell metabolism as seen in viral and some bacterial infections. Second is a toxin associated with the microorganism may adversely affect the tissues or organs of the host. We have studied the host damage by any parasite or by any pathogen, and one of them was toxin. And toxins are divided into exo and endotoxins. So now we are going to deal with these exo and endotoxins in detail. Two types of toxins called exotoxins and endotoxins are associated with bacteria. First are exotoxins. These are produced inside the cell and diffuse out into the surrounding environment. They are produced by both gram-positive and gram-negative bacteria. They are extremely toxic and are responsible for the serious effects of certain disease. For example, the toxin produced by Clostridium tetani, the causal organism of tetanus. It is neurotoxic and causes severe muscular spasms due to impairment of neural control. Examples of other toxins identified are necrotoxin causing tissue damage, enterotoxin causing intestinal damage, and hemolysin causing hemolysis of erythrocytes. Next one is our endotoxins. These are lipopolysaccharide protein complexes associated mainly with the cell envelope of gram-negative bacteria, while in the case of exotoxins, these were associated with gram-positive and gram-negative bacteria both. They are responsible for the general non-specific toxic and pyrogenic reactions common to all organisms in this group, that is in the gram-negative bacteria. The specific toxic reactions for different pathogenic gram-negative bacteria are due to the production of a toxin in vivo. Organisms of interest in this group are those causing cholera, plague, typhoid and paratyphoid fever and also whooping cough. So guys, thank you very much for watching this session. You can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and on LinkedIn. You can also subscribe ITLS Academy on YouTube and you can get it on from Google Play Store. Thank you very much again for watching this session.